So already we know about the plates as well as four-plate methods. So advantage in this one is possibilities. Sometimes each technique has their own advantage and on disadvantage will be there. So shall I move ahead? Next one is related to serial dilution. Already you have this again the serial dilution is generally performed when we do a serial dilution. Observe. And if you are keeping this plates on the longer incubations, there may be a chance that some organisms are having the growth rate very less. The chance to grow them, it will be available. It is like that you are in a class total. Sometimes I may ask, okay, some few candidates will start to say many things. Others are waiting. It is not a meaning that they are not knowing. Okay, but they are waiting for if others is not there. If it is being asked, then they will also going to reply. Okay. Here let's see. Some are the slow runner, or they are having the growth cell division is the time consuming. Sometimes the 120 minutes or higher time is there. We are not able to identify. So that is the main our second target. That those who are slow runners, they also can able to grow over it. When it is in higher populations, it may mask the growth of the others. It is like that. And going to be certain cookies here. Okay. Also some items is there. Everyone is start to learn. Those who are able to catch the other answer, there is no problem. No need to move in a rush. So here the rush will be there. The opportunity for growth of the various will be there. So serial dilutions. How we can do the serial dilutions? And what's the most important points to remember while doing the serial dilution is what? Can anyone say? What is the basic steps? Here you can see 9 plus 1 9 plus 1 9 plus 1. Okay. What's the basic important step one has to remember and recall during this step? Can anyone say? Let's look at this. Okay, I will take number 1 and 8 into 9. So it became total 10. Once again, I will take number 1 and 1. So it became Okay, it is like spreading. Okay, yes. This time. So if you are taking the one minute, half an hour will be go there, and will be wasted of time. Okay, so on understanding what was this one here, we are going to take with so what lesson. Within the lesson also we are going to take on a next or maybe on subsequent day. Second or one day you are going to also. What was the basic? Once there, let me call the thing just to visualize that if for same experimentation I may have asked you how you will perform, and one important step is required to consistently identify see the pictures or try to think that what I will do, how I will. Yeah. Specific step. Few steps are there. It's essentially not being taken. It's only provided important is it possible when you are asking one what should I use? What can you use? And then so shall I use the water or shall I use the liquid water? Water. Just I think you here. So network may not. Here is I have given 9 field. Okay, I have used the matrix here. Okay, smaller matrix here are for 10 plus plus plus. I have already given this. Here I use 9 field. Also, sterile. I have used this source. Next up is a core system between then which important essential step? Okay, how you will do serial treatment. That is the main question. In this process, we go what we study. There is also I think but in the all we are going to take one another from the going to transfer from there. We say okay, continuous data which we so we became what is I. We are not going to take from our original one another moving current, one moving current, one we goes. Such process will be continued. But in this process, only I must see this process only. Some steps you think about it. What I am telling, take one transfer this one now you will transfer once again transfer. Okay. In this process, take care very important points. Just think about it. It's always important. So one important part, level. You may write 110, 100, right? Like those. Everything is one of the another. Then you next thing. That is calculation cannot do it. That is not that not the guru which are that is very object. Right? It is not yeah. Volume. Volume you have to manage. Okay. Yeah, it is showing you. Yeah. 9 ml, you is given to you. So now that character is off. Okay. You are using the bracket to transfer to 1. That's what I was saying. But we haven't thought about. No one has told. No one has told. Why am I when you are doing the first step, I always ask you to start your biology. Right? Same principle is going to apply here. That you will take the 1 ml. Okay. Now, you are going to do the same thing. Number of microbes may going to increase. So you have to change the pace in each step. That was so very basic. It is nothing. Okay, it is nothing to say about it. Only people are very practical. And when they are doing the practically and understood the phenomena, only you have to know the facts about it. That time visualize the things what you are going to do once there. And coming here, not to just make you understand, make you learn. Learn it and with reason. Right. So it changes. Small basic step and we never given much attention to it. Have you thought anyone other than me? Yes. Always everyone should say that. But the normal thing is just forget. So each step we have changed the case. Okay. Which maybe not written in your protocol posture, never. That is why I'm telling you people, while writing the journals, you may write the protocols, afterward, write the notes. Okay. Write the notes according to you, the what you have learned during your different. Some of the people have different mid semester one also. So in the same lectures. Okay, same question. Can be asked again in 4 marks. Right now it is there enough. Another area can be asked in the few 4 or 6 mark, 8 mark patterns may be there. Something may be asked. But so we have to take care. I hope you are not going to forget it at least. Here we can do two things. One by one. First, afterward, from the same class, okay, respecting number flag, we may take about not one, maybe 1 minute. Start to and so it may be difficult to do twice the things, to do it better. Do a dilution continuously. Okay, you may go for the dilutions continuously. Afterwards, from each place, respective flag, you may transfer. This number is representing the dimensions. Here, when you are taking one to nine, this is 10 dimensions of the original. When you are taking the 1 and transferring here, that is 100 times dimensions. That is so much. This is 100 times dimensions. Once we take 1 and 9, this became 1000. Then goes from 10,000 1x. If you are having the 150 time, you may go and goes. I hope your question is clear, but sometimes I'm not able to understand some. So it's a time sense. So 10 achieve one x. Sometimes I have here water tank. You may put 1 drop of microbes over it, take some sample, the 1 to 1 lakhs time dilutions. But it is you have to use that water tank. When we are really just 2 samples, you can do it. So thousands to lakhs time dilutions we have to do. So I know how it's clear serial dilution, the concept of the serial dilutions. Important step in this handling microbial conditions, decontamination is required, handling with the sanitizer, everything is obvious. But the important part to handle it is few basic steps that you have to remember and record. Yeah, another important second question. Are you going to go for water or you can go for the nutrient broths for dilution? Between here you are when you have to do dilution, right? It is for a fraction of a minute, maybe 4 or 5 minutes there. Within 5 minutes, even though if you are using the nutrient broth, bacteria or the microbial population is not going to grow at a larger level. Or it may happen, but it is even for even time. Here you are doing dilutions, here for a hours to hours. It may be 5 or 15 minutes, and you are diluting. Again, if you go to the water, you are using the water, then also cell is not going to harm. Okay, you can use either water or a nutrient loss for a dilution. Anything could be good. So sometimes it is you may have the questions, but it may be understood why. If you are using the nutrient loss, then also they are not going to increase in that much number. If you are using the water, they are not going to die. Okay, so either of the our feasibility and availability of the water or the nutrient loss. Now clear? Anybody have any questions? Yeah. What could be the advantage and disadvantage of this method? Require more things I hope its point is clear. Shall I move ahead for our next topic in this line? Yeah, same thing is something is shown in different forms. Okay, here they have shown the case. After yeah, this lady, I think what someone is showing about.
Hello, I'm Dr. Carla Field. In this video, I will explain the serial dilution process and demonstrate how to perform dilutions when using the Microbiologics ePower product. Dilution is the process of making a solution weaker or less concentrated. In microbiology, serial dilutions or log dilutions are used to decrease a bacterial population to a required concentration. The purpose is to achieve an appropriate concentration for a specific test method or to achieve a countable agar plate. A single log dilution is a 10-fold dilution, which means the concentration is decreased by a multiple of 10. To complete a 10-fold dilution, the ratio must be 1 to 10. The 1 in the ratio represents the amount of sample added, and the 10 represents the total size of the final sample. For instance, a sample size of 1 milliliter would be added to 9 milliliters of diluent to equal a total of 10 milliliters. Before we can move on to multiple dilutions, it is important to understand decimal numbers and scientific notation. Decimal numbers can be converted to scientific notation by moving the decimal place the same number of places as the exponential number. In this example, if we start with 100 and move the decimal 2 places to the left, the final result will be 1.0 times 10 to the second. We use multiple dilutions to decrease the sample concentration by multiple logs. Here is an example. If the concentration starts at 35,000 CFU per milliliter, or 10 to the fourth, and 35 CFU per milliliter is the target concentration, we would do 3 serial dilutions. We would take 1 milliliter from the initial tube and place it in 9 milliliters.
So
this would
equal
about
what?
3,500
when you are
CFU per milliliter.
Colony forming
or 10
unit.
to the
What is the
third.
colony forming unit?
Another dilution.
Individual cell is able to develop the
concentration
colony.
to
It's called colony
350
forming
CFU
unit.
per
Maybe
milliliter,
suppose
or 10
this
to the
much
second.
number of the
And the
cells are
final
there for
dilution would
when you are
equal
transferring, so it
35
became 10 times
CFU per mil,
dilutions.
our target concentration.
I can give some questions, for example.
Now I will show you how to perform a multiple dilution series with Microbiology
For the dilution itself,
ePower E7. First,
you may go for
label
the dilutions,
plates with
or
the
second
dilution
maybe a
factor.
serial dilution.
Next, using sterile forceps,
I have
place 1
colony
pellet
forming unit.
into 10 milliliters of
This
pre-warmed phosphate
10
buffer.
raised to 2 per ml. CFU, colony forming unit. That means each individual cell
Vortex the
having the capacity to
microorganism
develop one
suspension
colony.
to achieve a
It's called colony
homogeneous
forming
suspension.
unit. Okay, I want to get this diluted to 1
Pipette 1
CFU per
milliliter
ml.
from
How
the
you can
suspension
proceed?
into 9 milliliters of phosphate buffer.
What she has shown
Repeat
here
this
also.
process until all
Colony
the
forming
tubes
35,000
have
was
been
there.
diluted.
10 raised to power 4. then diluted, then diluted.
Be
then
sure
going
to
on.
hold your pipette vertically to achieve maximum accuracy.
So they have used some plastic waste. There are the plates. C has already numbered. Let's see this documentary completely. Then I may give some questions.
Repeat this process until all the tubes have been diluted.
What C is doing, can you see? It's called the vertex. Okay, cannot mix in this way. Okay, vertex is there. It's just rotating. So over there, if you are going to keep, it will mix your sample. C is using the cap tubes. Caps lock tubes.
Plate
Tubes are there.
1 milliliter from each tube. Use a spreader to distribute the inoculum evenly.
Yes. C is using plastic. They are already available, sterile. The sterile spreader are available. For the documentary purpose he is showing, it is not the good way. So otherwise, it will take about 1 hour to complete this task, to show this document. Just for the sake of things, what is the steps are there. Now you are understanding also that what someone is doing is right or wrong, right? Just by seeing, you can see. So just think
Place the
that some
plates
person
in
is
an
asking the
incubator
basic questions to
overnight.
you. He is already about 10-15 years
These
experienced
are
guy
our
is there.
plates after the
Just
overnight
while
incubation.
interview,
These
they may
results
understood
demonstrate why it is necessary to do
that
serial
what you have done.
dilutions.
Otherwise, if we
Most
ask
of
for
these
some
gave
demo,
us either a
that
full line of
some few
bacterial
things is
growth
given to you,
or too
can you
numerous
do
to
it?
count colonies.
Suppose these are the
Our
two
final
burners. Can you
dilution
do?
gave us an
So once
easily
you start to
countable
open the
plate.
plates and close the plates, they come to know that how
For
prudent
more
and
technical
good you are in
information,
your
visit us
things.
online or contact our technical support team.
Okay. Yes, ma'am has that. Okay, ma'am has completed her work. Okay, fine. Now the question for me is here. I have how much? 10 raised to 2 colony forming unit per ml. I want to get 1 CFU per ml. How should I proceed? Okay. Okay. So I may go for 1 gem 10 dilution. Okay, first dilutions I am doing 1 gem 10. How much CFU will be there then? 10 CFU per ml. I am going for the second dilution. And that time I'm doing 1 gem 10 dilutions and I will get 1 CFU per ml. Can everyone able to understand what I have done? First dilutions. Original is what? 10 raised to 2. So when I'm doing the 1 gen 10, I have taken the 1 ml from here, added into 9 ml water or a nutrient broth. So resulting dilutions is going to become from 10 raised to 2 to it's became 10 raised to power 1 colony forming unit per ml. Okay, again I'm doing the dilutions. So their dilution is going to give 1 CFU per ml. Understood everyone? Okay. If I give some another example, just write it down. Then after I may give the another's. Now, क्या रे वरी खबर नहीं શું હતું પણ આ પછી આ ફોરેન ના લોકો કદાચ કઈક એ લોકો ત્યાં બીજી રીતે કરતા હોય પાઇપ પેટ છે એની આગળ કઈ અહીંયા અરે
Next, using sterile forceps, place 1 pellet into 10 milliliters of pre-warmed phosphate buffer.
It is the they have
Vortex the microorganism suspension to achieve a
they have used
homogeneous
the
suspension.
one E. coli directly. When you are working in experimental lab, I have told that sometimes we may go for transport the
Pipette
media, right?
1
Over
milliliter
there,
from the
they are showing
suspension
that not
into
bacterial
9
culture they have taken.
milliliters of
Just the
phosphate
bacterial
buffer.
flecks are there.
Repeat this process
When you are doing
until
some
all
experiment
the tubes have been
and the
diluted.
tubes has been kept. Okay, so just centrifuge it.
Be sure
Just
to hold
centrifuge
your
and then
pipette
you may
vertically
sterile
to
it. It
achieve
became powder form,
maximum
lyophilization.
accuracy.
It is a lyophilized E. coli powder. Metabolically, they remain active. I think discussed during the preservations, right? Microbial preservations. We have to
Repeat this
maintain the cell in a way
process
that they are
until
not going
all
to
the
die.
tubes have been diluted.
Okay, now write it down this in your diaries. I will give about 5 to 6 different questions to you. You need to clear it. Okay, write it down here. Yes, I may request you people to have the one diaries to write something. It is now really difficult in online sessions and offline sessions. I need to request, right?
Plate 1 milliliter from each tube.
Okay, next questions. Just write it down step by step in your.
Use
This
a
one is the
spreader
original
to distribute the inoculum evenly.
and desired. How will going for the dilution? First dilution, second dilutions. What I have written. That step I'm looking that you are going to write. Fine. So original is a 10 raised to power 8 colony forming unit. I am looking for 10 raised to 2 CFU per ml. My first question. Second question.
Place the plates in an incubator overnight. These are our plates after the overnight incubation. These results demonstrate why it is necessary to do serial dilutions. Most of these gave us either a full line of bacterial growth or too numerous to count colonies. Our final dilution gave us an easily countable plate. For more technical information, visit us online or contact our technical support team.
Just do only 4 questions. I think it will be more than enough for an hour, half an hour to complete. What I have written? Colony for original and desired. 10 raised to power 8. First question, second, third, and fourth. 10 raised to power 8 colony forming unit is there. You have to dilute it. That is a desired sample diluted. I'm looking. That's power 10 raised to power 2. So how many times I need to go for? Here, one important part will be come on the next stage when you are going to calculate it. You will able to remember and recall. You are going to follow this. Okay. Maybe you are going to do 1 gem 10 dilution sometimes. Otherwise, you have to change the dilution factor. Okay. The number as you start to calculate, you will come to know that why and how. Here you have taken the 1 and 10 times it's diluted. So you are going to write the number as 10 raised to 1. Then you are going for 10, or the number is decreasing, I have written some magic number. The magic is totally different. એવું તાન થોડુંક દૂર કર દો. રજા જ આપી પડશે ને. Yes, I know. Let mathematical magics may start. Some people may able to complete, other may not. How many guys are good into maths will come to know. Yeah, I'm looking for one class test, maybe in આ કરાવું કરાવી દો. Let's try with one. Yeah, regarding the class test I'm telling. Okay, in a class test, most probably and next week I will plan. Okay, about certain different questions related to this or the previously what we have done, or some related to theoretical or practical questions I will ask. Okay, that will be, you have to solve it and to submit me back. I will give the dates. Some maybe some example from similar to it. Okay, half an hour will be given to you. I think it will be more than enough on that time. So some questions already we have done. First related to earlier we have gone forward. I think for growth curve. From the growth curve we have some calculations we have done, right? Second, we have started to do this. Or else I may give or ask the questions related to how to do media preparations for so and so percentage or others. Okay, so in our next class tomorrow, I think you have distributed the numbers of the group. Fine. So tomorrow come with some calculations. I'm going to check during our class. So on a Thursday from the morning time, we'll go in a lab to perform it. So tomorrow, bring that chemicals, whatever the group wise is their status. Okay. Now the first question, 10 raised to power 8 CFU per ml is there, and you are desired it 10 raised to 2. When I'm doing a dilutions, okay, 10 raised to power 8 to here I'm looking for the 1 gen 10. 1 gen 10 dilutions, if I'm doing, the power is going to reduce 10 raised to 7. Okay, again from this first dilution, second dilution, 1 gen 10, it is going to become 10 raised to 6. Third dilution, it's going to become 10 raised to 5. Number 4, 1 gen 10, it may going to 10 raised to power 4. So I will need how much tube? 6 tube. What I have done, take the 1 ml. 1 plus 9 ml. So it is became 1 gen 10 dilutions. Initially it was 10 raised to 8. When I'm diluting, it became 10 times diluted. Once again, I'm taking the 1 ml from 8 plus 9 ml, I'm going to 8. So dilutions it became how much? 10 raised to 6. So step by step, I'm decreasing their dilution. 6, 5, 4, 3, 2. My desired number has came 10 raised to 2 CFU per ml. So I will take about 0.1 ml from it and going to pour on a plate. Okay, either pour or spreading can be done. So my basic question is what? To get this number. So you need to, if you are going to do this experimental part, you have to do 6 times dilution. Understood? Just write it down, then we'll go for the next question. Just I have given the 4. Let's see how everyone is going to do. So, okay, one just I will come. Okay, huh? No, first from the first one orders, dilution is started. From here itself. you have taken from the original 1 ml is culture, 10 time dilution. That is a 9 ml water is there. You have mixed in 9. So it became diluted 10 times. When you are taking the 1 ml, here it is, one tube is there. It's original. I have another tube. I have taken the 1 ml from here to there. Okay, here it is what? 9 ml water was earlier available. I have taken the 1 ml and I have transferred. In a 9 plus 1, it became 10 ml total. So original population was 10 raised to power 8. It's decreased to 10 raised to power 7. It became 10 times diluted. So now from this, if I'm going to take 1 ml and going to dilute with 9, then the power is going to rest down 10 raised to 6. So it will be repeated. And ultimately, I may reach up to 10 raised to 2 when I'm doing the 6th time that dilution. Understood? I hope. શું હતું? એ ભગવાન, બહુ મોટો ક્લાસ છે, આમ દોડી દોડીને થક્યું છે. લખતા જાઓ, પછી સેકન્ડ થર્ડ આપણે કરશું. It's the mathematical fundamental things. People may able to remember, sometimes may not. But once you have perfectly done, then you will easily able to perform. Eh? Ama, I do. Just I will come. Just do it. એવડા મોટા નંબર નાખી દીધા છે ને કે ગોઈતા જ રાખશે. ઢૂંઢતે રહી જાઓ. Just let me just, I may sometimes forget, so it's became easier for me. પાછા next time મારે કાલે આજે નો થાય તો કાલે સુધીમાં ભૂલાઈ જાય આંકડા. Yes. આ તો થઈ ગયું હવે. Very nice.
અને 46 નું ડાયલ્યુશન હોય ને ત્યારે 2 ml per 1 per 10.
How you come to know about 6 times?
8 minus 2.
Okay.
6.
Fine.
તો એવી રીતે 48 minus 2 એટલે 46 થાય. પણ 45 સુધી આપણે એમ નમ કરવાનું. 46 માં આપણે એને ડબલ કરી નાખવાનું.
શું કામ?
કારણ કે અહીંયા 35 છે, અહીંયા 70 આપણે.
Okay.
એટલે આપણે 2 ml લીધું, 2 ml પર 10 મતલબ 2 ml લઈ અને પછી એને ડાયલ્યુટ કરી 10 એટલે 35 into 2 એટલે 70 થઈ જાય. હા એટલે થઈ જાય ને.
Here it will be, last number is total volume sold, always.
આપણે ડાયલ્યુટ કરી
What you are going to do here?
આમાં શ્યોર નથી પણ 4 છે અહીંયા 2 એટલે 2 ટાઈમ ડાયલ્યુટ કર્યું પછી 125 માંથી 5 કરું એટલે 25 નો ગેટ આવે 25 into 5 એટલે 125 એટલે આપણે 25
નથી બોલ છે. કરો કરો ભાઈ.
આટલું કર્યું.
હા હા.
હા પછી આગળ કરી એકવાર
તમે ભેગું કરો કઈક હવે આમ. શું મોટા? હા.
35 છે બરાબર 1:10 નો રેશિયો લીધો. હવે 35 છે 35 10
જો અહીંયા શું થયું? આ આંકડો ઘટાડવાનો પેલા તો. આને ભૂલી જા. પેલા આના ઉપર વિચાર. 48 છે. અહીંયા શું થશે? 47. બરોબર છે. ઈ ક્યારે થયું? 10⁻1 કર્યું એટલે 1 ઘટાડ્યું ત્યારે. આપણે શું કરવું છે? 2 કરવા છે. બરોબર છે. ઈ ક્યારે થાય? કે જો અહીંયા વિચાર, આ બેનો ટોટલ કેટલો થયો? 47. આપણે કરવો છે કેટલો? બરોબર છે. અહીંયાનો ડિફરન્સ કર 48 - 47 એટલે 1. હવે મારે આ કરવું છે તો શું કરીશ તો થાય? 10⁻46 1 જેમ 46 ટાઈમ. તો મારે શું થઈ જશે? થઈ ગયો ટોટલ 48 માંથી 1 ગયું 47, 48 માં 46 ગયું એટલે 10⁻². તો ઈ કેટલું રિઝલ્ટ આપશે મને? 35 into 10⁻⁴. અહીંયા તો 2 આ આવશે. તો મારે જોતું હતું 70. વધી ગયું, આ ઘટી ગયું મારે. તો હું એનાથી એક સ્ટેપ પહેલા તો આગળમાં જઈશ, 46 સુધી નહીં લઈ જાય, એને શું કરું 45 લઈ લઉં, 10⁻⁴. તો શું થશે 35 into 10⁻². અહીંયાથી જશે 3 CFU પર. હવેનું પાછું વિચારો, હવે મારે 70 કરવું તો કેમ કરીશ? પેલું સ્ટેપ શીખવાડો. કર્યું તે સમજાય ને? હા યસ.
આમાંથી 2:10 નો રેશિયો.
અચ્છા.
46 જેમાં 185
Okay. But I am looking for 70. 46 time dilutions કરી નાખ્યું તો રિઝલ્ટિંગ આન્સર શું આવશે ત્યાં? 35 into 10⁻² આવી જશે. I am looking for this. બે દાખલા એકાર આપ્યા છે ભાઈ, એટલે રિવર્સ ગેરમાં થોડુંક હાલવું પડશે. સાચું કર્યું છે, નિયર બાય છે, આણે ઈ જ કર્યું. શું કે ના કરો તો કઈક બેઠા એમ તો જીઓ એટલું આપણે તૈયાર કરીશું, બસ તમને પાઇપેટ આપવામાં આવશે. એટલે 1 ml 1 માઇક્રોલીટર. હા 1. પણ શું એટલે ml માઇક્રોલીટર શું યુનિટ? હા 0.04 એટલે 4 માઇક્રોલીટર જ છે ને. હા 4 માઇક્રોલીટર તો આપશું, તો 0.5 માઇક્રોલીટર સુધી કરવા દઈશ એવી પાઇપેટ આપીશ તમને. ઓ ત્રીજામાં પહોંચી ગયા ભાઈ. નથી સમજાતું, એટલે તો એવા સરસ આંકડા આપી દીધા કે અઠવાડિયા સુધી મૈથા જ રાખતો હતો. Yes man, going on then? Not yet. So plan for to which? Yes. Yet not done. I know it's somewhat difficult. Mathematical reasoning is required a lot. Oh. Yes, very nice. So it's of 6 factor is coming 6. Yeah, 6 times. So you have done for 6. Very good. Okay, so how many times I need to dilute? And another. Here it will be coming what? 35 into 10⁻⁴. But I need 70. Yeah, 70 into 10⁻² column forming unit. You are near to right. Okay, then two-step process is there, not on one step. You're right, near to right. ઓકે, ક્યાંથી આવ્યો? અચ્છા પાડોશી આપ્યો. હા બરોબર. અરે બધા આપણે બધા we are in a group. હોય હા વાંધો નહીં, કરો તમે તમારે. આપણે આપણો 70 into 10⁻² થવા દો. હમ
17 to 10 raised to 6 બેને મિક્સ કરી
અહીંયા 6 નથી, અહીંયા શું છે?
એટલે એમ નહીં
અચ્છા, અચ્છા.
અને મલ્ટીપ્લાય 2 કરીએ ને
હા, હા.
આનાથી 4 times value કરી નાખીએ એટલે 70 to 10
અચ્છા. તમે શું કહો છો? બેને ભેગા કરો છો, પણ એને પર ml માં કેટલું થાય? તમારી આગળ 5 રૂપિયા છે, 5 rupees પર પર્સન. આની પાસે 5 rupees પર પર્સન છે. બેને ભેગા કરી દીધા, 10 rupees પર 2 person. પર પર્સન ઇઝ રિમેન 5 rupees. એટલે આ કોન્સેપ્ટમાં ભેગું ઈ નથી થતું. ઈ માયલું ન થયું. હે આ એટલે નહીં ભેગું થયું. એટલે જ ખાલી આ 4-5 દાખલા દઈ દીધા ને. તમે તમારે અગર ઓનલાઈન હોય, ગૂગલ હોય, જુગલ જે હોય ને, ભેગું જ ન થાય. જેને આવડે એ જ કરી શકે. મારી ટેસ્ટ એવી જ હશે, ઓપન બુક ટેસ્ટ જેવું જ મોજ કરો તમે તમારે, બરોબર. આવડતું હશે ની દાખલા ગણીને કરી હશે, માર્ક્સ લઈ હશે, પછી નકર આપણે બેસશું આરામથી. એટલા બધા ટફ નહીં પૂછ્યું. માય પ્લેઝર. યસ. No. It is you have taken 1 ml. Okay. So first you are taking 1 ml. So it's going to remain as number 35 only. This power is reducing. This number is going to remain as such. It's not going to change. Yeah, that was my desire. Yes, that I am looking for this number. I have kept always 10⁻² as my per ml 1 solution. So first time I thought that I will go for 10⁻². So it is 1 into multiplied 10⁻². Second time I thought that no, I will go for the 70 into power 2. So always my desire is changing. Yeah, it is totally different. So this is mathematically derivations. You have to learn it. So I hope tomorrow you people will come with solved only 4 questions. Because time is આ તો થઈ ગયો. જો આને તમારે કરાવવાની જરૂર. હવે ટાઈમ નથી, 9:30 થઈ ગયા છે. આ તો ભણવા માટે ઉત્સાહી છે બધા. નકર ઓલો એમ થાય આનો છે જવા દો. આજે ઉલટું થઈ ગયું. થિંક હિયર આ ટેબલ છે મારે કેવું પડશે હટાવી દો. આ દર વખતે મને પડવાની બહુ બીક લાગે. આ જૂના વાલા હું ક્રાઇસ્ટમાં હતો ને 3-4 તો પડી ગયો તો એટલે ખબર ન હોય મગજમાં, પછી ન્યા જ હોય કે એન્ડ આગળ ઉભા હજી એમ હોય કઈ પડ્યું ટેબલ. અહીંયા what we have seen here, just I'm going to solve only one, at least one. Okay, so here from this the reasoning will come. Okay, just everyone keep attention here if possible. What we have done here, 10⁸ to power 2 I'm looking. Okay, so how many times it is required to do a dilution? 6 times. Okay, so when I'm writing here 10⁸, what generally we are writing here? It's a 1 gen 10 raised to power 1 I can write, so it's become 10⁻⁷. Directly if I'm looking, so it's a 1, 1, 1, 1, 1 and 1. If I'm going to add all together, it's become how much? 6. So I'm writing directly. It's not easily, it cannot be possible because it required 1 a tank to do it. Okay, larger tank to do it. But mathematically, I'm just writing how much is 10 raised to power 6. So it's became how much? From the beginning, it's a 10 raised to 8. If I'm diluting from 10 raised to 8, it's became 10 raised to power 2. Okay, so it's my desire to write it down. Okay, in one step I have written, same thing I have written over here. Okay, what I have done, 1, 1, 1, 1, 1, I know about it. So it became 10 raised to 6. So what is the basic things you can understand from here? If it's 10 raised to 8, when I'm starting to write, when I'm writing is a 1 raised to, something is understood. My desire is 10 raised to 2. Okay, so I can write in this way, 8 minus 2 is equal to 6. Okay, so 10 raised to 8, I'm my original 1 gem, 10 raised to power 6. If I'm going, then colony forming unit per ml, I will get this. I hope understood. Many people will not easily able to understand because it's a mathematical thing. So this is understood till this. Okay, now my part I'm looking for this. If I'm writing same 35 into 10 raised to 48 is my desire. Okay, I'm looking for in this format only. Here I'm writing original number, whatever it is there. Okay, something I don't know, but I'm looking for, here it is suppose 30, 70. I'm initially putting as 35. Suppose this one is mine required. What I can do, 48 minus 2. So it will coming as how much? 46. Understood everyone? સમજાય છે? It became 46. Now this one is going to happen, but I know that I have done one mistake. What mistake? Not a mistake, but my desired sir has about this. So there I have to do a certain change. So this one is became my CFU per ml, but I was looking for something different. So from higher we can go for the dilutions, but from lower to cannot. Once you're having the buttermilk, you have added the water. Okay, so it is mixed, or if the milk is there, you have 1 liter milk is there, many people are there, you may have added half glass water into it while preparing the tea. It's become diluted. Tea is prepared. You cannot take out the water from it. You need to boil. You can boil it. There it's possible. Here you cannot. Once it is there. So reverse one step we have to move. Now my desired is something different here. So I will go for 35 into 10 raised to 48 to 35 into 48 is 10 something. I will not directly put this. Okay, I will may put 35 into 10 raised to power 3. I will wait back, coming back, and if I'm putting 48, so it will be how much? 45 times. It will be 48 minus 3 is equal to 45. Now my number is here. Colony forming unit per ml has came this. I will take now work on this. Okay, how I will move? Now I'm looking for how much? 70. Okay, how much it is amount? It's double. If I'm going to dilute directly, then it's a problematic. So what I may do, if I'm going to take 1 ml out of it, yes, we may going to take amount will be a double. So 35 into 10 raised to power 3. If I'm looking, then I'm not going to take here this number. We may go for the dilutions in a way that, yeah, what we have writing here, it's a 1 gem in this format. Okay, so here the number is going to change. 70 into 10 raised to power 2 CFU per ml. So what I will do, I may go for taking the 2 ml. That's right. I may go for taking 2 mls. And total is going to how much? Going to create 10 itself. So what basic thing is, 2 ml is my inoculum plus 8 ml is my either distilled water or nutrient cloth. 10 times dilution is there. Okay, but originally what I have done, I have taken not 1, I have taken 2 ml. So twice the quantity has came, but it is diluted till this. So here my number is 10 itself, right? But the number is taken not 1 ml. Total is became 10 ml. Be careful while doing the dilutions. Make up 10 ml. One word. Many a time it is written. I think in biochemistry, many a time sirs or someone is telling the same thing maybe. You have to make up the volume 10 ml. So the dilutions will be considered in this process. It is diluted. Okay. And the dilutions factor is coming what? 2 ml plus H. Okay, so it is going to give the 70 10 raised to power. Column for me. Huh? Yeah, total number of dilutions, how much count? This one is number 45. Okay, 45 સુધીનું કર્યું અને 46મું કરો છો 46 only, but this is in different format. Understood? I hope. Ha. Yes. Answer, yeah. Number of the dilutions. Number of dilutions you have performed is 46 times. If you are following 111, here, yeah, I haven't told. Okay, that is the 1 ml is concept is there. Okay, I'm not going to make it complex, but it is just assume that you are taking always 1 ml or 2 ml. So till 1 to 45, you are taking 1 ml, you are diluting and diluting, right? And second time you are doing, when you are performing, you are going to 2 ml, make up a 10. So what you are going to make the last factor, that is your dilutions factor. Understood? I hope. Now, pardon? Okay, try to understand from your colleagues first. I hope, because time is also not there. That's why I told that maybe some able to understood, some may not. હવે તો થઈ જ જાય બધું હમણાં, પછી આંકડા જ છે. આ સ્ટેપમાં જ કોમ્પ્લેક્સ કર્યું હતું. Clear? Majority, some may have their own way to write it down. Okay, the difference just it is what you are going to do. હા વાંધો નહીં, અટેન્ડન્સ લખી દેજો. સારું હાલો, પછી બીજું કર્યા રાખશું. Yeah, put your attendance. કાલે વેનસડે છે? નહીં, થર્સડે ના જ કરશું. કાલે આપણે કાલના ક્લાસની અંદર ગ્રુપ વાઇઝ ની કેલ્ક્યુલેશન્સ ની ચેક કરીશું. થર્સડે શું કરવું નહીં.